Hello and welcome to this evening's Science for All Seasons talk. The Science for All Seasons talks give you a chance to explore hot topics in genomics and biology with leading experts from the Broad Institute. My name is Tom Ulrich, and I'm the Associate Director for Science Communications at the Broad. And it's my pleasure to welcome all of you here this evening and to introduce our two speakers for tonight, Benjamin Neale and Bronwyn MacInnis. Ben is the co-director of our program in medical and population genetics, and Bronwyn is the director of our pathogen genomic surveillance program and our infectious disease and microbiome program. Ben and Bronwyn will talk about COVID-19 through the lens of two different genomes, that of the SARS-CoV-2 virus and our own, and about what those genomes are teaching us about how our bodies respond to the virus and about viral evolution and spread. Before we begin, I'd like to take a moment to note that tonight's talk is also the 2021 Eliana Hechter Memorial Lecture, delivered in honor and memory of Eliana, who was a gifted MD PhD student and a member of the Broad community. And I would like to welcome her family and loved ones who are in attendance with us this evening. And with that, Ben, over to you.
Thanks, Tom. And it's a distinct honor to be delivering at least half of the Eliana Hechter Memorial Lecture. I knew Eliana in passing. We met a few times outside of Eric Lander's office, and I think we shared a similar sort of passion for science and curiosity about the world, and perhaps a stronger commitment to the mathematical and logical and insightful aspects, and less enthusiasm for the outright computational things that sometimes come along for the job. But it truly is an honor, and I think Eliana really was, completely and utterly enthralled by genomics and how genomics and genetics can be used to learn things about the world. And I think the kind of two talks today are a fitting tribute to that legacy. So genomics, COVID-19, and us. We'll be kind of walking you through both, as Tom said, the human genome side and the viral genome side. To start, I'll give you a little introduction to myself. So my undergrad, I completed at the University of Chicago and Virginia Commonwealth University, focused in kind of genetics more broadly. At graduate school, I did a PhD in psychiatric genetics, and indeed have been focused primarily on psychiatric genetics throughout most of my career, not only in grad school and the postdoc, but also as an assistant and now associate professor at Harvard Medical School, Mass General Hospital, and the Broad and the Stanley Center. And you might kind of be wondering why a psychiatric geneticist is talking to you about COVID host genetics. And, one of the things about being a human geneticist is that for the last, say, 5 or 6 years, maybe 10 years, there have been concerted efforts to try and bring various parts of the human genetics community together. And a lot of the work I did in psychiatry and continue to do in psychiatry is characterized by kind of this consortium science model, this idea of a whole bunch of different scientists working together. And even though I focus on psychiatry, I'm generally interested in genetics and generally interested in what we can learn about genetics and about human health and disease by using genetics. And so launched the Global Biobank Meta-Analysis Initiative with Mark Daly, Kristen Willer, and Wei Zhu about, I think, 3 years ago now, sort of before the pandemic. I don't know, time gets a little bit fuzzy in these pandemic times. And the kind of express purpose of the Global Biobank Meta-Analysis Initiative is to try and aggregate, genetic analysis and results of common human diseases to learn more about why people get sick and how that all operates. And so this kind of framework, I think, provided a very natural entry into this fundamental question about COVID. And, at this point, I think we all know someone that's gotten sick with COVID. Many of us know people that have died because of COVID, that the kind of landscape of variation that people have in terms of what happens when they get infected is quite diverse. And so as a geneticist, a very kind of natural approach to this question is to try and, understand why some people are getting really sick or dying, whereas others have no symptoms. That's like the kind of heart of the matter. And as a geneticist, the way that we sort of approach this, and this is a kind of very slimmed down human genetics approach in a nutshell sort of worldview, we start with recruiting a cohort of humans, or maybe multiple different cohorts of humans, and got little emojis here to give you the idea that this is really trying to capture large chunks of the population where possible. And indeed, the Global Biobank Meta-Analysis Initiative is a whole bunch of active cohorts where human genetic information has been collected and can be analyzed if you can collect it, not only the genetic information, but also the phenotypic information. So we need to know, we need to identify the people, we need to collect the genetic information in those people, and then collect whether or not they have had COVID. If they have had COVID, how bad was their COVID? What symptoms did they have? Were they, admitted to the ICU? Were they hospitalized? Were they, did they die? All of these questions are very much in frame and kind of looking at genetic variation, the genetic variation that every single one of us carries can help give us some clues about why people might be really sick if they get COVID or might have to go on a ventilator. And so we kind of collect the humans, then we collect the genetic information and the phenotypic information or the trait information, and then we analyze. And this is a picture of a kind of research compute center, a kind of data center. My lab also developed something called HAIL, which is a kind of general use software platform for the analysis of genetic data. That really is targeted at scaling up to the very large sample sizes that I think that we certainly need in order to be able to really identify which genetic variants are conferring risk to the various outcomes that we're interested in studying. Okay, so that's the sort of backdrop of human genetics in a nutshell. This COVID Host Genetics Initiative, how it started, well, March 16th in 2020, Andrea Gana, who had completed his postdoc in my lab and is now on the faculty at the Finnish Institute of Molecular Medicine, working with Mark Daly, who's the head of the FIM and also kind of longtime close collaborator and institute member of the Broad, launched this COVID-19 Host Genetics Initiative. We started with a tweet out to the world. And in response to that tweet, we've now kind of aggregated and put together over, 3,300 researchers worth of efforts across 61 studies in 25 different countries. This represents one of the largest genome-wide association studies ever done. And all a genome-wide association study is doing is it's analyzing not just one genetic variant across the genome, but it's trying to identify and analyze every single genetic variant an individual has that's common enough to be captured on the kind of typical genotyping arrays that we use for these sorts of things. Okay, so that's kind of the landscape. And as the pandemic has evolved and changed and shaped our world in all of these different ways, so too has the science. We've been continually and committed to generating that kind of analysis of comparing the genotype with whether or not someone gets COVID or how bad their COVID is. And we've proceeded across a series of freezes that we release generally to the entire community, to the world's community, once we are satisfied that they are of sufficient quality, that they are reasonably trustworthy. And these freezes have grown from, a kind of initial studies of only 1,600 people with COVID to now a meta-analysis where we have 125,000 individuals, genetic information, and a reported positive COVID test in the analysis. So this is truly unprecedented. I mean, it normally would take a decade to do this sort of thing, and we've accomplished it in a year, a little over a year. And I've never seen anything like that in my career before, and I kind of am of the suspicion that I never will see anything like it again. And let's hope that that's the case with respect to these kinds of pandemics. And so I'm going to walk you through a kind of series of figures to give you a flavor of where the cohorts are coming from and how there are sort of different sample sizes with different kinds of questions. So the three main analysis questions that we focus on, the three main sets of phenotypes that we're looking at are over here in this lotus plot on the right-hand side. So that kind of orange color reflects critically ill with COVID-19. So those are individuals that have either died because they have COVID or have required sort of intubation or some additional, some assistance in their breathing. So it's really trying to capture the sort of sickest of the sick. hospitalized with COVID is similar, but it doesn't require the same level of granular detail on what procedures were performed to an individual if they were admitted to a hospital. So we don't require that there was evidence of intubation or something like that in terms of the identification of the hospitalized with COVID data set. And then this third one in the kind of teal color at the top is just individuals reporting whether or not they have had a SARS-CoV-2 infection. So just a positive test. And so that opens up a variety of different kinds of cohorts, particularly things like ongoing research studies where the participants fill out surveys, as well as things like the direct-to-consumer companies like 23andMe and the like that also contributed to this effort. So we've also got on the left-hand side here the kind of effective sample size, which is a way of reflecting, how many cases there are and how many controls they are in a kind of even-handed manner, because some of the studies are more traditional, recruit a bunch of individuals that are in the ICU and then try and match them to controls. And so that's a very targeted case control approach, in contrast to some of these more epidemiological style cohorts where you're just, asking 100,000, 200,000 people whether or not they've had COVID or not, or whether or not they've been hospitalized with COVID or not, and kind of recruiting your phenotypic information that way. This is an international effort. So here's the kind of world map, and each of the different labeled panels here represent different contributing sites for the overall meta-analysis that I'll talk to you about through the rest of this talk. And if we zoom in to each of these regions, so over here, this A box is represented down here is this A box, you see, a lot of the kinds of academic medical centers and biomedical research, focal points in both the US and Europe as grossly overrepresented in this kind of figure. So we see like Boston is one of those, or the Bay Area is another one of those, as you might imagine. But with COVID, it's a global disease. This is, it's everywhere. And the sort of good thing about this effort is that it is amongst the most international that we have ever achieved in terms of trying to understand the genetic basis of any human disease. The bad thing about this effort is if we kind of break apart the effective sample size by sort of ethnicity or self-reported ancestry, what you end up seeing is that the overwhelming majority of this cohort is still of individuals of European ancestry, and most of the cohorts are coming from Europe or the US. And that's just another sign that we have a lot of work to do to close the diversity gap in genomics research on all sides of the equation. And that is something that I think many of us share is an important next step as we continue to work to advance our understanding, not only of COVID, but of human disease more generally. So some take-home messages just to orient you about what are some of the most important things that we think that are coming out of this. First and foremost, there are clear genetic effects. That means that genetic variation is clearly and significantly contributing to whether or not an individual will have a very severe experience with COVID or whether they will have asymptomatic disease. No real presentation. The only way that they would know that they had COVID was if they had managed to get tested at the point in time. Many of these associations are not apparently shared with what we know about many other viral or other infectious diseases at this point in time. And that's kind of interesting. I mean, I think there's a lot about the immune system that we don't fully understand in terms of how kind of response to pathogen or, kind of fending off infection operates across the full diversity of all of the bugs that are out there in the big wide world. But that specificity is, I think, interesting because it might teach us something about how susceptibility works in this context. And kind of related to this question of susceptibility to infection versus the severity of the infection, there are some genetic effects that clearly overlap between, kind of susceptibility and severity, and some things that look really, really specific to things like disease progression and are really not related to whether or not you're having susceptibility, but are actually much more related to how sick you're going to kind of get. And so that, I think, reinforces this idea that as having a virus establish an infection in an individual is different from what course that virus's time in that individual takes. And I think that this is, kind of maybe one of the hardest parts of infectious disease to really study and understand, and the genetics is certainly supporting that there is some potential gap there. And then finally, I'll walk through the kind of vignettes on the biology much more, deeply. But immune response genes really matter. And what we know about, the kind of immune system, and what we know about lung biology, and what we know about the genetic landscape of those things is, kind of reinforcing that this kind of approach can teach us the kinds of insights that we would like to learn in order to better treat COVID, because I think if, our treatments got a lot better, that would change our response to the pandemic, but also maybe think about other ways of prevention. Okay, so let's dig into the results now to go a little bit deeper. So what I've got laid out here are a subset of the chromosomes in humans, so 23 chromosome pairs per human, and we've only, kind of narrowed down and zeroed in on the set of chromosomes where there are clear association findings. Again, we've got this sort of lotus style color coding for the critically ill with COVID-19 versus the reported SARS-CoV-2 infection versus the hospitalized analysis. And so the colored in leaf of the lotus plot is the kind of significant association. So that's like what that particular locus is significantly associated to. And as you can even see on this kind of slide, that there are some things that are relatively specific to hospitalization, like this TMEM65 locus, whereas there are other regions where the kind of pattern of association kind of runs across the full range of phenotypes that we've got collected. Under each one of the kind of sample, kind of association descriptions, there are our efforts to try and identify and show which gene or genes might be involved in that. And I'll kind of talk about some of these in a little bit more detail now. So let's kind of dive in with the first one here, this FOXP4 association. I think one of the most critical things about this particular association is that the genetic variant is markedly more common in individuals of Asian and Hispanic and kind of Latino populations. And so without really a kind of global effort and really including and incorporating diversity into our association analyses and into our genomics work more broadly, we really wouldn't have been able to identify this genetic variant. The kind of frequency of this variant is way, way, way too low in European ancestry individuals. And so there's just very little chance that we would have learned that this particular variant is associated to SARS-CoV-2. And particularly the kind of reported infection and the kind of hospitalized, it's kind of splitting across both. For what it's worth, the same genetic variant that's popping up around this region of FOXP4 is also associated with other lung-related phenotypes, so lung cancer and interstitial lung disease risk. So again, pointing to this idea that lung function is a critical aspect of how bad your course of COVID illness is. And there's even a likely mechanism of action based on what we know about this particular genetic variant that has been studied before because of the prior associations to lung disease, that it likely results in increased expression of FOXP4. And for whatever reason, increased expression of FOXP4 is yielding increased risk for reported infection and hospitalization. Next up is a locus on chromosome 19, and this one's a little bit more complicated. So there are a bunch of different genes in this region of the genome where the genetic association is being found, where we kind of try and, identify that signal that we think is pretty believable. The kind of drilling down into this association and carefully walking through what the genetic variants are in the region and what we know about the patterns of association. This localizes to a gene called TYK2 or TYK2, which and a specific missense variant. Okay, and so what a missense variant is it's a mutation that changes the amino acid structure of a protein. So instead of one amino acid, you have a different amino acid in the sequence of amino acids that make up your protein. And so this is a kind of, relatively speaking, big biological effect for a genetic variant to have. And as a result is something that we've studied a fair amount as a community for quite a long time because of its role in reducing risk to many autoimmune diseases. And indeed, there is a TYK2 inhibitor that is being developed by, I think, Bristol Myers Squibb as a treatment to psoriasis, so an autoimmune disease there. I think it's psoriasis, wouldn't totally quote me on that. But unfortunately, that's in the opposite direction for the risk for severe COVID disease. And so this is, again, reinforcing this idea that in autoimmunity and immune, kind of function in general, there is this push me, pull you between an increase in your risk for autoimmune disease and then the degree to which you are good at fighting off infection or fighting off severe infection and those sorts of things. And so there's this kind of balance between infection response and autoimmunity biologically. And then, the kind of next up here is this gene SLC6A20, which is significantly associated really with infection susceptibility and not with hospitalization or getting critically ill with COVID. And, the kind of very plausible reason that this seems like an important gene is that this binds to ACE2, which is thought to be the host receptor for the SARS-CoV-2 virus. And so, the kind of, as it's binding directly to the receptor where the virus is getting into the cells, it seems like a pretty plausible biological candidate in addition to the kind of genetic association. And here again, we see that it's actually specific to infection rather than critical illness or hospitalization. All right, so that's a kind of taste of some of the biological results and associations that are sort of cropping up. As I sort of alluded to a little bit earlier, we're committed to this open science idea and this open science framework where as soon as we have results that we trust, we release them to the world for everyone to use. And indeed, our latest data freeze is just posted last month. And we've moved from the kind of 50,000 cases to 125,000 cases, including some new associations that are popping up that I think have some pretty readily interpretable kinds of biological stories around them, including genetic variation around the gene ACE2, which is the receptor for the virus in the first place, as well as the association to this SFTPD gene, which is a lung surfactant gene that is involved in responding to respiratory pathogens. So more associations coming, more potential insights and starting points for thinking about how we understand both infection and severe illness in the context of COVID. So with that, I'll close and just point you to the COVID19hg.org website, where you can read about everything that we're doing. You can access all of our webinars and all of the kind of communications that we've done within our community in order to accelerate the science and really understand as much as we can about this devastating illness. And with that, I'll say thank you for your attention, and I think turn the floor over to Bronwyn.
Thanks, Ben. I'll just take a second to get my video loaded. And great talk. I actually started my work in infectious disease looking at why some people are more susceptible to malaria than others, and pivoted into looking at the pathogen genomics, which I'm excited to tell you all about today, the work that we're doing on COVID. It is a tremendous honor, really, to share the work of an incredible team at the Broad and our network around the world who have been using the power of genomics, something that is core to our work at the Broad, to support public health response to the COVID pandemic. First, just briefly about me, I lead the Broad's pathogen genomic surveillance program, and I am as a scientist, a genomic epidemiologist, which basically means that I take the age-old science of epidemiology, things like mapping case numbers and symptoms and deaths, like you can see here in this iconic map, locating the Broad Street pump as the source of cholera infection in London in the 18th century. To understand how infectious diseases spread and cause disease. But we're adding in a whole new data type, and that's really the magic of the genomic part of genomic epidemiology, using the complete sequence of the pathogen causing the infection. So in this case, the SARS-CoV-2 virus that causes COVID, to understand how cases are connected and how the enemy, the virus, is evolving its arsenal as it spreads in response to our public health interventions. So briefly, we use this data, the genomic data, in multiple ways for outbreak response and in worst case scenario, pandemic response, including in the very beginning to use the data to identify the causative agent of disease, when someone presents with symptoms like fever or respiratory infection that could be caused by many things, how do we know what it is if it's something we've never seen before? We go right to the genome to determine that. And we in fact did this for COVID back in January 2020. We then use that genetic information to guide diagnostic development, and the diagnostic that's still used globally as the gold standard, the qPCR test, is essentially based on knowing the genetic sequence of the virus itself. And then a lot of what I'm going to focus on comes after those critical initial steps, using genomic data for surveillance, surveying for known or emerging variants with important functional effects, things that will change the way that we want to try to combat the spread of disease. And then more focally for epidemiology, to understand in a targeted way how cases are connected, kind of like contact tracing 2.0, where we ask not humans where they've been and who they've been in contact with when they're ill, but actually ask the pathogen or the virus itself. And then finally, as a forward-looking thing, ultimately we see this data as influencing clinical medicine. We're not quite there yet, but we're close to the point where sequencing the genome of a pathogen, a virus causing disease in an individual patient can ultimately alter the course of how it's treated clinically in terms of what therapies and even potentially what vaccines are administered. I just wanted to back up and say that although, the idea of genomic surveillance and asking kind of what variant of infection someone might have seems like a fairly new thing in our common conversations. It's actually been around for quite a while, and I've been privileged to be part of the community that's really developed this technology and approach for multiple pathogens, viruses, and other critters, and to use that to help global infectious diseases, including malaria, back almost a decade ago now, the Ebola outbreak in 2014 that the Broad Viral Genomics Group was deeply involved in, and more recently the spread of Zika during the last global pandemic in 2016. But of course, none of these really compare to what we've experienced with COVID, and that's what I'm here to tell you about. So I am here, in fact, because the Broad has launched a major genomic surveillance program for COVID on the back of its massive COVID testing program. As you may know, the Broad is best known for its efforts in genome sequencing. A lot of that has to do with sequencing the genomes of humans, as Ben just told us about. But when COVID came, it pivoted its tremendous resources and robotics and ability to handle massive numbers of samples to meet the critical need for diagnostic testing, and ultimately provided about 20% of the tests that were done across the country, largely serving Massachusetts and the Northeast. And when the need for genomic surveillance emerged with the emergence of variants of concern earlier this year, we added this dimension to the Broad's response to the pandemic, such that positive samples from the testing program moved into our genomic surveillance pipeline. This meant that these positive tests, when they were found, got shuttled into our sequencing process. The virus was isolated and prepared for sequencing, and then sequenced using the Illumina platform. And then with that genomic data, it gets sent to our computational biologists and computer scientists who wrangle it and analyze it to understand what it's telling us about how the pathogen, how the virus is evolving and spreading. And then we share that information with our public health colleagues in the state and at the CDC. So this effort contributes to what is an incredible global effort in genomic surveillance for COVID that has to date generated in excess of 2.5 million SARS-CoV-2 genomes. This is just a stunning amount of data that is orders of magnitude greater than anything that we've ever had for any previous outbreak or pandemic. And I think is really setting a new standard for how we will do public health and respond to outbreaks and pandemics of the future. Here I'm showing just a representation of that 2.5 million genome data set. It's of publicly available data on the GISAID database. On the left, you see a phylogenetic tree. This is how we often look at the data, showing the evolution of different viral lineages, different kind of sub-flavors of COVID over time, where each dot is a sequenced virus collected from an infected person from a positive diagnostic test, colored by lineage, or more commonly known as variant. This is only about 0.2% of the total data that's available. And there's a lot going on here. You don't need to worry about the details, but what I just wanted to show you first of all is that over time, starting early in 2020 and proceeding through to July of this year, there's been just a massive uptick in the number of genomes that we've been sequencing at the Broad and around the world in response to the variants of concern that emerged earlier this year. You can also see that there are many viral variants, not just the few that we talk about like Alpha and Delta most commonly now, but actually many viral lineages and only a few that are really emerging with mutations that may confer functional consequences that are more threatening. Most of the mutations that differentiate viruses are benign and just give us a genetic fingerprint that we can track. And you can also see that there have been, this explosion of variants that we are concerned about, including most well-known, I'd say, the Alpha variant first identified in the UK, here in blue, formerly known as B117. And of course, the Delta variant first identified in India, that is previously known, as B617.2, but fortunately we have these more simple names, Alpha and Delta, to keep track of them now. On the right, you can just see the global representation of these viruses and all of the data that's poured in from sites really around the world. You can see that there's a concentration of the Alpha lineage in the UK and Europe. You can see the Gamma lineage first identified in Brazil, in red, predominantly in South America, and in India, you can see a predominance of the Delta variant, but of course, most of these, in particular Delta, has really spread globally now. And what do we use the data for? Oh, oops. What are we looking for, I guess, in this, when we sequence these genomes? We're looking for viruses that obviously spread more readily, for viruses that can cause more severe disease, can alter the detection of our diagnostics, reduce the efficacy of our therapies, and of course, most pressing now, evade immunity, in particular by our vaccines. So just taking a moment now to zoom in on what we mean really by the viral genome of SARS-CoV-2 and its variants. It's really striking to me that such a tiny piece of genetic material, just about 30,000 bases, just about a millionth the size of the human genome, is causing such devastation around the world. But what I really want to just share with you is that the genome is made up of about 12 genes, and one in particular, the spike gene, that is the focus really of a lot of attention. It's not the only gene that we're focusing on, but it's one that our vaccines are generated against, and that really holds the key to unlock entry into the human host through the ACE2 receptor that Ben told us about. Variants or lineages, as we know them, are defined by mutations that can occur across the genome, but predominantly we are interested we tend to focus on mutations that occur in spike because they do tend to have the most functional consequences. And here on the lower panels, you can see a model of what the spike protein actually looks like, where mutations that are commonly known to exist are shown in red and orange colors here. And you can just imagine that these mutations can affect the way that the protein interacts with the ACE2 receptor to kind of enter the door into the human host cells. Sorry, I'm trying to advance here, but it's not working. Okay. So And in terms of the variants themselves, we are, of course, tracking both known and emerging variants, but the ones that have really captured our attention and are the focus of a lot of science and public health work around the world include alpha, the beta variant first identified in South Africa, gamma first identified in Brazil, and delta, as we mentioned, in India. And these variants have different phenotypes, different ways of affecting disease, of affecting spread and affecting immune escape, but they all do this more or less to some degree in a way that means that we need to focus resources on trying to understand their spread and understand the impact that they're having, especially with respect to spread and vaccine escape. And delta obviously is the one that we're paying the most attention to at the moment. Its ability to spread is on the order of about 2 times the spread of the 2 or more times really the spread of the original SARS-CoV-2 virus. And it has been reported to both cause more severe disease and to affect immune escape. This slide really just shows just how much more effective the Delta variant, known as 617.2 here, is at spreading. We use the term growth rate here as a more kind of scientifically accurate measure of what we're actually describing, but we think it's a proxy for the incredible ability of this variant to transmit. So what you can see here is that the Delta variant 617.2 is substantially better at spreading than B117, and of course, many of the previous lineages that came before it. And interestingly, I think you can see that variants of concern and variants of interest that we're tracking are increasing over time. And it's important for us to survey continuously, not just for these known threats, but also for what of these that are kind of coming up on the radar may be something like the next Delta variant or the next Alpha variant. And that's a big part of the work that we do, is both to continue to track known threats, but also continue to survey for emerging threats. There are currently about 1,200, almost 1,300 known SARS-CoV-2 lineages. So there's a lot of work to do in real time to be monitoring these for things that we need to be paying attention to going forward. And so what are we seeing? Here I'm showing you 2 plots, one of across the US and one in the US Health and Human Services Region 1, which is the region that we're serving in the Northeast. Showing the proportion of infections across these regions of a given variant of concern or variant of interest over time. The plots start in April, and as you can see, just as recently as April, the B117 or Alpha variant was the predominant variant, in both the US-wide and in our region, although you can see there are some differences in the proportions of variants that we see more locally versus at the national scale. This B117 variant, if we could go further back in time, had risen dramatically from being just trace amounts just a few weeks before to reaching about 70% of prevalence of cases across the US and about 50% in the Northeast. But then over the subsequent weeks, from really just the end of April through to just last week or so, you can see that the B1.617.2, the Delta variant, has increased from just trace amounts to now comprising on the order of about 80 or more % of infections across the US, depending on the region that you're in. So this just goes to show you just how transmissible, how readily this Delta variant can spread, and is obviously a large focus of our work and of the work of our community who's doing this across the country. But we're not only looking at the state scale or at the region or national scale. We're also working closely with local public health officials to support our state and local investigations into outbreak, cluster investigations, and other public health concerns. And most recently, this is focused on a cluster investigation that we're working with the state of Massachusetts on in Cape Cod, focused in Provincetown. And this has been reported in the media. There's several stories. I think this is a very kind of real-time investigation that we're a part of. The state epidemiologists have identified about 883 cluster-associated cases. 500 or so of these reside within Massachusetts, and about 200 are actually within Provincetown. And the others were visiting Provincetown over the 4th of July weekend and subsequent weeks. And so it was really a moment where there was, we were finally feeling like we could relax just a little bit. Vaccination was high, cases were lower, and people wanted to celebrate, understandably. But what we've seen in the situation is that despite high levels of vaccination across this community, there has been significant infections. And many of those infections are in fact symptomatic, which is one of the most concerning things about this outbreak. In fact, we know that the vaccines were designed to protect against, in particular, severe disease. So we're not so concerned when we hear about necessarily about vaccine breakthrough infections, but hearing that these are actually causing symptomatic cases has kind of sounded the alarm to understand what's going on here. So of course, we sequenced as many of these cases as we can, and this work is ongoing, but so far we're seeing that about 90% of the sequenced cases are in fact the Delta variant. One of the first questions that the public health officials wanted to know, and of course we as scientists and genomic epidemiologists wanted to know, was, is this a new variant? Was it Delta? Was it some kind of funky new version of Delta that seemed to be even more able to spread and able to cause disease? But we're happy to report that so far, no, that doesn't seem to be the case. Of course, this is a work in progress, but so far it looks like a fairly standard version of the Delta variant that we've been tracking around the world. And interestingly, we've also seen that, there were multiple introductions, multiple sources of these Delta viruses into the region. So even though in terms of the functional mutations that are consequential, it looks like a typical kind of Delta infections, they do have marker mutations that enable us to determine that they came from different sources. So this suggests that there wasn't just one major super spreading event necessarily that has led to these infections, but that multiple sources have seeded infections in this community during this time period and have led to kind of outbreaks of different sizes. Some are bigger and some are smaller, and that's work that is ongoing. And finally, I think, although we don't have concrete data to show this conclusively yet, I think it does raise the hypothesis that there is spread between vaccinated individuals, given at this point, just based on the fact that so many people were vaccinated and so many people got infected. So that's one of the questions that we're following now, using the genomic data from the viruses and combining with the kind of traditional epidemiological contact tracing to determine, can we see formal links between vaccinated individuals where transmission has happened? So more about that coming soon. But that being said, it's important to note that our vaccines are still effective, and in particular for fully vaccinated individuals. I think it's safe to say that the vaccines, while not being perfect, are absolutely still our best defense against COVID infection and COVID disease for ourselves and to protect the wider community. And here you can see data from multiple sources kind of amalgamated, showing that in particular, when you have 2 doses of either the Pfizer, in this case, the AstraZeneca vaccine, there is continued protection, although some decrease of protection from symptomatic infection. And this is expected because we know that the measure of effectiveness of these vaccines, as I said, was really against severe infection. And when we look at that on the right, you can see that for severe infection in both Pfizer and AstraZeneca vaccines against both the Alpha and the Delta variant, there does continue to be substantial protection. So I think it's important to note that even when you're hearing about these cases of vaccine breakthrough infections and in some cases symptomatic disease, these are rare. And that is really exemplified here in data from the CDC that was released last week, where they looked across the entire US population, where an incredible 161 million Americans have been fully vaccinated. And there's been only, to that point, about 6,000 breakthrough infections requiring hospitalization, so more severe sickness, representing 0.003% of these vaccine breakthrough cases. The percentage of vaccine breakthrough infections, at least in Massachusetts, even still that remains small. It's only about 0.1% of even just at the infection level that are vaccine breakthroughs. So despite the fact that we're seeing some cause for concern in this cluster in Cape Cod at the moment, I think it's really a signal that we need to continue to be vigilant and continue to monitor what's going on. But in the meantime, our vaccines are still our best defense. So given that, and given the fact that the world has access to these vaccines, who is still at risk? And I think the answer is pretty obvious, but just want to put it in concrete terms here with these data, that it's the pockets, both on the domestic kind of across the country and around the world, where vaccination rates, for whatever reason, whether it's accessibility or uptake, remain the biggest risk. And I think it's safe to say that while there's great progress in many areas of the country and the world, it's these pockets, and in some cases kind of gaping holes in some of the lower resource countries around the world, that really we need to focus our resources and energy on getting vaccinations levels up across the globe to make sure that we protect these communities and really protect ourselves too. Because as long as the virus is continuing to spread and continuing to evolve, these mutations, we really don't know yet all the tricks it has up its sleeve. And so the things like Delta and potentially even more risky forms of the virus will continue likely to emerge until we have reached a level of immunity through natural infection and hopefully mainly through vaccination that protects us all. So with that, I just wanted to leave you with a few questions. There are, of course, many questions. I hope that you have questions for us, but some of the ones that we're continuing to look at together with our colleagues in public health and scientists around the world include, what's happening in kids, how quickly can we get vaccines that are approved for use in kids, and how much of a threat is Delta to younger age groups? Can it spread between vaccinated individuals? As I mentioned, I think we're starting to see some data that suggests that if you are infected, even if you're vaccinated, you can probably continue to spread, but we'll continue to monitor that one. Of course, the question about should we be wearing masks, I'm happy to go into a bit more detail on that one. What about needing boosters or vaccines, and will new and potentially more threatening variants continue to emerge? I think the answer is it's a bit of an arms race, and we will need to do everything we can to get the world vaccinated to protect against the threat of new variants. And so finally, just a huge thanks to everyone involved, including in particular the amazing team in the genomics platform for all the testing and sequencing that they're doing, and our colleagues in the data scientists platform who help us wrangle the data, all of our external partners, funders, and the incredible community around the world that's really stepped up to bring this new technology to bear on the COVID pandemic. And with that, I'm happy to take questions. Thanks.
Well, thank you so much, Bronwyn, and thank you so much, Ben. And thank you to everybody in the audience who've been sending their questions in. We've got some great questions that have come in so far. Hoping maybe Ben and Bronwyn can stay a little past our 6 o'clock time, just so that we can get through as many of them as we can. The first one that I want to put to Ben actually was kind of around not so much the science, but the process with COVID HDI. You said it basically started a year ago or so with a tweet. How did you and your collaborators get it up and running and churning out data so quickly?
So, sort of two ways. I think the sort of first and foremost approach was to kind of capitalize on existing cohorts where the genetic data is already available, and sort of working with that as a sort of, as people were getting infected, that was then being reported in their health record, and then that health record gets shared in an anonymized way with researchers that have access to the genetic information, in a kind of secure, controlled access kind of environment. And so that enabled, particularly in the context of, one of the large scale, really publicly available epidemiological cohorts of the UK Biobank, sort of real-time analysis of, as people were getting sick, they were being, brought into hospitals, and then that hospital electronic record information was then being pushed over to the human geneticist to analyze that kind of data. And so that actually, you can kind of think about how, kind of human genomic surveillance of outbreaks or epidemics or any sort of major public health might be conducted in exactly the same way in the future. That if we had a sort of fully integrated healthcare system with all of this information electronic and available, and then linked to the genotype in much larger populations, it would really streamline and accelerate this even further. Then on the other side, with kind of acute pandemic, severe disease like this, scientists immediately started to recruit patients and immediately tried to start to build the cohorts and the sample sizes to really get the sickest of the sick and compare them to people that seem to slip through the kind of COVID infection landscape with a relatively mild or even asymptomatic course. And so both of these approaches of like focusing on the individuals that are really sick, but also leveraging existing cohorts with available genomic information has really facilitated this whole work. And I think it'd be impossible to do that without a decade plus of consortium science and human genetics across the world working together in these kinds of ways to build the overall meta-analytic activities that we are, kind of revealing the insights that we think are important for COVID more generally.
Bronwyn, I've got a question for you. And actually, this is from me, not from the audience, but it's something that struck me while you were speaking. You talked about a lot of different kinds of variants, but really it came down to two flavors of variant: variants of interest and variants of concern. And I've heard these terms in the news as well over the last 18 months or so. What do those terms mean? What's the difference?
So the difference is basically just how much of a threat we think they are. It's a construct that has been set by the global kind of public health community, including the CDC and the WHO, to try to triage all of these lineages that we were starting to track as they were coming at us, and where we should focus attention most. So I think as soon as a variant emerges, or a variant lineage that seems to have maybe some kind of effect that we think we need to pay attention to, it makes it onto the variant of interest list. And that's typically looking at increased spread, maybe in one area, or potentially immune evasion or more severe disease. And as those initial findings kind of replicate, or those observations are seen in other areas of the world, or in other communities, or in different kind of assays and ways that we're measuring these things, the threat level increases, and the decision when to move a variant from the variant of interest to a variant of concern is made actually at the country level, and the WHO makes a decision across kind of the global threats. but it's been one of the many interesting dimensions of this work to watch how different countries decide what's a variant of concern in their country and when they'll actually kind of make the call to elevate a particular variant lineage to the concern level. And I'll say there's also I guess it's kind of like a highest level threat, a variant of high consequence category, where the variant actually has been shown to kind of render interventions, less useful or much less useful. And so far, we don't have any variants in that category. So that's an important thing to know. But we're obviously keeping our eye on Delta and whether it will elevate to that level.
And actually, building off of that answer, another one of the audience members was asking if we have any evidence or inklings or anything about the effectiveness of the vaccines that we have today against some of the other variants that are out there, like Gamma or Lambda. Have you seen anything about that?
Yeah, so most of the studies, there's been so many studies, it's hard to synthesize all the data. Certainly for the Gamma variant, there is some evidence that it can evade immunity, both natural immunity and vaccine-mediated immunity. That is the variant first identified in Brazil. For Lambda, I don't know of data on that yet. That's a more recent kind of variant that we're tracking. It's only just arrived in the US. I saw that reported yesterday. So I think it's something that we're tracking, but so far the prevalence of those variants, at least in the US, is relatively low. So our vaccines should work again. We aren't seeing a massive spread of those across the US, fortunately.
Thank goodness so far. Ben, one of the audience members would like to know if someone has had their genome scanned by a commercial outfit like 23andMe, can they analyze their own data to see if they are carrying any of the risk variants that you've identified so far?
So I think that's possible with 23andMe. I think they enable you to download your own data, but I'm not absolutely certain about that. But you'd have to download your own data. And then even after having downloaded the data, the kind of starting genetic information that you get from a genotyping array is not every single common variant you have. We use something called imputation, which is a statistical procedure to sort of fill in the genetic variation that we didn't type directly based on reference population information about the patterns of genetic variation and how they correlate with one another, because there's a lot of correlation amongst genetic variants in human populations and in many other populations. So it's sort of, in principle, maybe feasible. I guess I would be remiss not to caution about how modest the effect sizes are for some of these genetic effects, right? Like they're not, really big effect sizes. They're pretty small. And so the kind of modest change in risk is not what I would describe as like hugely interpretable for an individual risk point of view. Like it's much more likely that like the amount of virus that you're exposed to, the variation in the virus that you're exposed to, and the, how many times you're exposed to the virus are going to have a lot more to do with whether or not you're going to get an infection than what is going on in your own genetic variation. And I think there's plenty of epidemiology to support those kinds of statements. But you could go and kind of have a look if you were curious, and you could kind of navigate your way through all of the bioinformatics I just described.
Okay. A question that I want to throw to the two of you. Long COVID has clearly become a concern among people who have been infected by the virus and have gone on to survive. Do you see anything in the viral genome or the human genome that correlates with long COVID or someone's risk of developing long COVID?
So speaking on the human side, we're not in the same place with long COVID that we are with COVID itself, but the COVID Host Genetics Initiative is spun up a long COVID work group. led by George Davey Smith and many others, trying to ask exactly that and answer exactly that question. What are the kind of genetic risk factors that give rise to long COVID, and how do we think about that as part of the pandemic? And also with that goal of trying to understand the sort of biological basis of long COVID, of why, and I think there are a lot of open questions about exactly what's going on with respect to long COVID that I'm not going to speculate on too much here. But I think what is important to sort of recognize is that it's not just the pandemic and the acute illness that we need to deal with as a society. It is the full range of consequences of the virus that we need to deal with as a society. And on the human genetic side, we're committed to not only continuing that work, because we recognize that this is, life-changing for at least a subset of individuals that have had COVID thus far. And so we think there is and will be, because there's, kind of genetic influences on most of the things in health and disease and medicine. And so we think that's a promising avenue to learn more about it, but I don't have anything to report for this question just yet.
Okay. Bronwyn, how about on the viral side? Have there been any signals that you've seen popping up as you and your colleagues look at the virus?
Not yet. We and others are certainly looking at this, not just at long COVID syndrome, I guess, more kind of at long-term infections, persistent infection, reinfection, and any viral kind of markers or associations there. I think, I would guess that has more to do with the host, if not the genetics, the immune system and other factors, as Ben was saying. But certainly a question to ask about whether there's anything about the virus at play there, and something that we're looking at.
One of our audience members was curious about the pressures on the virus to evolve and to form new variants. And I was wondering if you could talk about that for a little bit, Bronwyn.
Yeah, so I mean, the evolution of viruses is obviously a very normal thing. And I'd say there's probably like multiple factors at play. A lot of them have to do with the population that they're in. And so I'd say one is just the kind of, the more infections there are, the more cases when you do see a surge, when public health uptake or interventions are low and case counts are high, there's just more shots on goal for the virus to naturally mutate as it spreads from host to host, and for certain mutations that may confer some advantage to be selected for. The other, I'd say, kind of common or mechanism that accelerates the evolution of virus in a way is in infections in immunocompromised individuals, where the individual can't fight the infection and they end up being sick for a long time, being infected with active replicating virus for an extended period of time. And with that pressure of a kind of weak immune system, you're naturally going to select for fitter viruses, viruses that may develop mutations that enable it to kind of persist in despite an immune response that it encounters in another host when it transmits from that individual. So I think those are two of the kind of driving forces that are driving evolution, just large scale case numbers and infections and infections in immunocompromised individuals.
Ben, I want to take the question over to you as well, but with the little add-on, is there any role that human genomic diversity might play in viral evolution, as it comes across new populations of people and new sets of variants within the human genome?
I mean, it's a great question, Tom. I think it's worth flagging that one of the genetic associations is to something called the OAS gene cluster, which is our innate viral immunity mechanism. So, we've developed arms to fend off bugs in our evolutionary history. And there's genetic variation around those kinds of defenses that mean that the defenses are subtly different between individuals. And so while we may, well, basically everyone has the OAS gene cluster, exactly how it works might be subtly different across individuals. And these are the kinds of clues that I guess we're trying to discover in the human genetics inquiry in general, is to try and further understand what that variation in the population looks like and what those individual differences really are. And so we can think at a species level about some of these things, but we can also think at like kind of the individual level. In terms of other kinds of viruses or other sorts of analyses along these sorts of lines, there are certainly plenty of documented instances of human genetic variation varying regionally, in particular because of the presence of specific pathogens or illnesses. And some of those defenses take the form of working through the immune system, but some of those defenses take the form of working through the system where the illness is being presented. And so it's not about the immune system, it's about the cells or the tissue that the kind of virus or bug will kind of take up residence in. And so, I think all of these things are in play, and I think there's just a huge amount of diversity in a lot of different ways that can give rise to some of these phenomena.
One question that's come in that it might be a little early in the in sort of the scope of the pandemic to be asking this, but I'll ask it anyways. Ben, so past major epidemics have sometimes left a mark on sort of like the human genome. Is certain people have done better in the face of a pandemic or not, and they pass on genes or variations that allow future populations or future people to also handle certain pandemics or certain viruses or bacteria better. Do you see any sign of, or do you have any thoughts about whether COVID might leave sort of its mark on the human species?
I mean, I think it's a good and open question. I think the kind of example that springs to mind is, CCR5 and the sort of protection that may be conferred with the, Black Death and the, protection that's observed for HIV. But at the same time, that same variant is associated with increased risk of infection of West Nile virus. So I think there are a lot of these, what I would describe as push me, pull yous in the game of immune surveillance and response, not only with respect to host, but also with respect to all of the bugs, right? Because it's not just one illness that we're going to be fending off at any given point in time. There are lots of circulating pathogens that in the population that we kind of deal with as a species. Is it leaving a kind of evolutionary selective mark? I mean, probably, but it's kind of a bit of speculation because it's got to relate to things like how many children people are having and like the age at which the kind of virus strikes. And so if most of the mortality is in the over 60s bunch, which is the case epidemiologically, the selective consequence might be a markedly different consideration than if a lot of if the mortality were equally distributed across the age range in the population. And so there are all of those kinds of factors that'll relate into the sort of how we think about natural selection and evolution with respect to the human genetic variation. So I'd be a little cautious in saying that there's going to be a kind of super huge, super strong signature of this, but you could imagine some subtle changes here and there that might emerge or might be boosted in allele frequency because it conferred a bit of protection to COVID.
Bronwyn, one of our viewers has a question about transmission by vaccinated individuals. Can you tell us what we know today about, especially people who are vaccinated but are facing the Delta variant? What do we know about transmission in that context?
Well, we're just, I'd say, at the point where we're beginning to have data to put together to understand how big of a threat this is. As I mentioned, in fact, this cluster that's started in Provincetown is in fact probably one of the first major data sets that might allow us to quickly kind of get a handle on how big the risk is of transmission between vaccinated individuals. I would say in principle, if you are infected and you have a substantial viral load from that infection, you could spread that infection. I don't think that we have evidence that would suggest otherwise. And now we're starting to see that, in fact, there's some indications, early indications out there that some of these vaccine breakthrough infections are in fact pretty high viral load. So if that's true, I think there's, I'd say, probably no reason in principle that you couldn't spread it to other individuals. And I think we'll see how that bears out in real data in the coming weeks, with our own data that we're generating here and that others are working on around the world.
Okay, well, we are well over time, so I want to ask one last question. And Bronwyn, I'm going to put you in the hot seat here. What advice do you have from a public health perspective for educating people who still have doubts about COVID's severity and spread?
Such a great question. I think vaccination is still the best tool we have. We are so lucky that that the scientific community and everyone who's worked downstream from that has been able to rally to develop these vaccines and get them distributed, loosely around the world. Obviously, still some big gaps, but and we need to do all we can to take that up. And I think it's probably preaching to the choir if you're still tuning into this session that you may be a believer in the power of vaccines, but I think that we can all be advocates for that. I think it's really those one-to-one interactions that you can have with folks in your own worlds who may be skeptical to encourage them to get vaccinated. I think that's an important job for all of us. And so we'll I think if we can do our piece there as a community, that's really what we can do. And then as individuals, I think we're seeing some change in guidance from public health just in the past day or so around masking. And I think, just from the data that we're seeing from this outbreak in Provincetown, being vaccinated, as we know, as was always kind of written on the box of these vaccines, they're not perfect. And we need to also be smart about our own behaviors. And it looks like, it's early days, but based on this and some other situations that are emerging, that close proximity indoors, in large crowds may still be a risk, even, in many times it may not, but under the unfortunate, wrong circumstances, the virus obviously can still spread between to people who are vaccinated. And so I think we need to kind of heed the guidance to continue to mask when that threat is higher, and just do what we can to prevent the spread, in particular to folks who aren't vaccinated for whatever reason, whether it's a choice or because they can't.
Well, thank you, Bronwyn, and thank you, Ben, both for all of your answers and for giving a wonderful talk. And thank you to everyone in the audience for joining us virtually this evening. We hope you enjoyed tonight's talk. And if you like what you heard, then consider watching other talks from the Science for All Seasons series. You can find them all at broadinstitute.org/SFAS. A recording of tonight's talk will be posted on our YouTube channel at youtube.com/user/Broad-Institute. To find out more about other live streamed events and new videos from the Broad Institute, consider subscribing. Tell your family and friends to subscribe as well. I hope you'll join us for our next Science for All Seasons event in October. Announcements will be coming soon about that. Thank you so much for joining this evening, and have a great night.